Hello, my name is Ximena Castro. I'm a PhD student at INCIBIO in Tucumán, Argentina, and I'll be presenting this work titled Proteomic Analysis of Elongated Alpaca Embryos. South American camelids are an important economic resource and cultural heritage for the high Andean populations. However, its production is limited by low reproductive rates and higher embryonic losses than other domestic species. One striking reproductive feature in South American camelids is that 98% of gestations occur in the left uterine horn. Therefore, this study aimed to characterize the embryo proteome to unravel the molecular basis of the maternal-embryo dialogue that leads to this particular implantation. In order to achieve that, 4 alpaca 15-day embryos were pulled into groups. Total proteins of each pool were isolated, fractionated in SDS page, and analyzed by liquid chromatography with tandem mass spectrometry. The spectra were analyzed with Proteome Discoverer software for protein identification and Blast2Go and InterProScan tools for their categorization. We also defined high and low proteins abundance groups based on the quantile density distribution of the exponentially modified proteins abundance index (MPI). Proteins in the top 20% quantile or the bottom 20% from the abundance values were categorized into high abundance and low abundance groups respectively and submitted to gene ontology enrichment analysis. A total of 519 proteins with at least 2 detected peptides were identified. Gene ontology analysis showed that most of the proteins were localized in the cytoplasm in association with the membrane or the endomembrane system. The main molecular functions were tagged as binding, catalytic activity, and structural molecule activity proteins. And regarding the biological processes, the main annotations were metabolism, biological regulation, response to stimulus, localization, development, signaling, and immune system processes. We believe that all of these annotations are consistent with a growing embryo and its preparation for the implantation. As I mentioned before, a gene ontology enrichment analysis was performed with high and low protein abundance groups. No enriched GeoIDs were found for the low abundance group. But in the high abundance group, the main annotations were intermediate filaments, polymeric cytoskeletal fiber, structural molecule activity, for example, ribosomal proteins, and apoptotic regulation. Getting these annotations within the most abundant proteins reflects again the intense metabolism of the 15 days alpaca embryo, which is elongating and growing. This work provides the first proteomic profile and functional in silico analysis of pre-implantation alpaca embryos, laying the foundations to better understand the maternal-embryo dialogue in South American camelids. If you want more information or have questions, please feel free to contact me at my email address. Thank you very much for your attention.